Hey everybody, this is Ms. McFadden, and I'm here to talk to you about DNA profiling and gel electrophoresis so you understand what it's used for and how we'll use it in the next couple of days in our classroom. So first, DNA profiling is a process that allows us to compare DNA sequences to help us understand relationships between organisms, the identity of particular people, or maybe even ancestry. Um, Almost all of our DNA is exactly the same from person to person. And if you think about it percentage wise, 99.9% of the DNA in your body is exactly the same as my DNA or as the person sitting next to you. However, when we think about how much DNA there is in our bodies, 0.1% is still a whole lot of differences among you and the person who's sitting next to you. And if we're thinking about how many base pairs that might be, how many letters A, T, C, and G, we're talking about maybe 3 million different letters that exist between you and the person sitting next to you. So even though it's small percentage-wise, it's really big when we think numbers. All of us have unique DNA. Every single person has unique DNA except for those who are identical twins. Their DNA sequences are exactly the same, which is why we call them identical twins. So when we're talking about DNA, what is the point of DNA profiling? Why do we do it, and what are the possible reasons that we might be involved in this? Generally speaking, we use DNA profiling to help us with criminal cases. They might help us, excuse me, resolve immigration arguments in terms of where did somebody originate from. They are also extremely important in determining paternity cases. So, who's baby daddy? And also, they help us identify remains from perhaps a crime scene or from some kind of a disaster that may have occurred, and bodies might need to be recovered. And then finally, we use them to identify ancestry or relatedness from individuals. Um, when we're thinking about DNA profiling, we can also think about some really specific cases that might give you some examples about how we use these things. One of the very first important cases of DNA profiling happened because there was a little boy who was supposedly the son of a British citizen. And he was supposed to be allowed to enter the country, but they had to prove that he was actually the son of a British citizen so that he could enter the country. And they used this DNA profiling to prove that his DNA matched the DNA of his father, who was in fact a British citizen. Another really important first case actually freed a man who had been held in custody for the rape of two schoolgirls. And when they analyzed the DNA from the crime scene and compared it to the suspect, those two did not match. So there's no way he could have committed the crime. There's also so many notorious cases where we've used this DNA profiling to figure out whether the DNA of a suspect matches what the DNA was recovered at a crime scene. There's another example in 1988. There was a suspect named Tommy Lee Andrews, and they actually matched the DNA that was left at these notorious rape cases to the suspect's blood cells to determine that he was actually the culprit. And so we use them often in sexual assault cases, and we actually compare the DNA from victims that we may be collected from victims to the DNA from suspects who maybe committed the crime. And if the DNA patterns appear the same on a gel, then we can pretty much conclude that that suspect is at fault. Lots of other reasons that we've used DNA profiling in the past. One of the most famous cases was comparing Dolly, remember the clone sheep? We took DNA from Dolly, the clone, and compared it to the ewe that donated the nucleus. And we knew they were exact clones because their DNA was exactly the same when run on a gel. There was a baby that was stranded in the tsunami in Sri Lanka, and they were able to take the DNA from the baby and determine the identity of that baby based on the DNA from parents who were looking for lost children. We've used it before to prove whether or not various leaders have had children in various different ways. And a famous case is this study about whether Thomas Jefferson actually fathered children with one of his slaves. And using DNA, we were able to prove that, in fact, he did. We've been able to use DNA profiling to help replace the use of dog tags in the military, because dog tags can easily get lost. And so if we can prove the existence of DNA from recovered bodies, then we know who of our fallen soldiers have actually fallen in battle, which has been pretty successful. And then we've actually been able to look at the differences in DNA fingerprints of families that maybe have these inherited diseases that we're trying to trace. So letting people know what their risks are for the inheritance pattern for these disorders. Okay, so what does this actually look like? This is a DNA fingerprint set from 4 sets of twins. And as you look at these, you should be able to tell which of these twins are actually identical twins and which ones are not. And just looking across here, identical twins will have the exact same, what we call banding patterns on the gels that we run. And if you look here at these 2 twins, they have the exact same strands in their DNA. And so do the twins in column C. They have the exact same DNA pattern. But if you look at the twins that are A and the twins that are D, you'll notice different banding patterns. And that's because these twins are still twins, but they're not identical twins. So that means mom had 2 separate eggs that were fertilized with 2 separate sperm from dad. Twins B and C were the same egg and sperm that were fertilized, but then split in development, and that's why they have identical DNA. And you can see the banding patterns here are pretty easy to identify in terms of which ones are identical and which ones aren't. Sorry, my slide doesn't want to move forward. Okay, so how else have we used this DNA tool? We use it often in wildlife population management, and the way that we do that is we look at how closely species are related to one another, because that can give us clues about whether these organisms have maybe been exposed to excessive inbreeding. And the closer their DNA banding patterns are, the more closely they're related. And that will help us start to manage species a little bit better. Cheetahs, you know, are really, really closely related. So if we were to take DNA from one cheetah and DNA from another cheetah and compare it, the banding patterns would look really, really similar. And that tells wildlife biologists that they need to introduce a little bit of diversity into the gene pool so that the population of cheetahs can continue to be successful and have enough genetic variety to succeed. The other thing that we use DNA for in wildlife management is determining if we're looking at 2 different species or if we're looking at the exact same species. Hey, I'm filming. I'm going to shut this. Sorry, a little bit of an interruption. Anyway, we can identify whether these 2 animals are distinct species or if they're the same species just based on how different their DNA is when we put it on a gel. And the other thing that we can do is we can identify animals that maybe have been illegally poached just by looking at the DNA that we collect from those carcasses. That'll tell us if these animals are endangered species or what type of species we're looking at. Because sometimes the remains from a poaching are hard to identify. So DNA helps us with that. So how do these DNA profiles actually work? Typically, when we are looking at DNA profiles, we're looking for an identical match. So we are hoping to find a DNA profile of someone that matches a sample that we already have. We can use a variety of different strategies to do that. One place that we often will get DNA samples from is from semen. And we'll take samples from semen and run that semen to help us determine if maybe the suspect from a sexual assault case actually committed the crime or not. We can also look at DNA from mom and dad and compare it to baby to see if that offspring is actually the offspring of these 2 parents. And the way that that works is that a baby's DNA banding pattern should be a combination of both mom and dad's banding pattern. It won't be unique. So it'll be some combination of mom and dad, but it won't be different from both of them, if that makes sense. We can also use DNA profiles to help us look at siblings, because siblings should have really similar DNA profiles because they're related to one another. So how do we use these profiles for identification? In order for us to do this for crime scene analysis, which is what we're going to be using this for, we have to have the bands in an unknown profile be an exact match for a positive identification. And so we're looking for the exact same individual based on these DNA fingerprints. If we're trying to identify a male from some kind of a semen sample, all bands in the sperm have to match the suspect's DNA profile, but not all the bands in the suspect's DNA profile will match with the sperm. And that's because each individual sperm only has half the DNA at a time. And that's just a kind of a side note. That's not really super important for you all. However, if we're trying to identify paternity or maternity, the bands of the baby have to be present in either mom or dad at all times. And that's kind of interesting. And we will do some practice with that eventually so that you can see that. Sorry. Okay. So here's an example of trying to figure out who this child's dad is. And if you take a look here, we've got two possible fathers and a mother who we know is the mother. And the way that we analyze this is we look at mom's DNA, and we can clearly see that the child has the same bands in some places as mom does. We should also look at dad's DNA and compare the banding patterns with dad. Now, each band that the child has needs to be either in mom's DNA or in dad's DNA. So the way that I look at a child's DNA is I take a look at the first band and I say, does mom have this band? And in this case, mom does not. So that means this band had to come from dad. And so we look at the two possibilities from dad. And it could be either male 1 or male 2 in this case. So I move on to the next band. This band looks like it came from mom, which makes a lot of sense. Then I move on to the third band. This band did not come from mom, so it had to come from dad. And as I'm looking at the possibilities, male number 1 clearly has this band in his DNA, but you'll notice that male number 2 does not. So that tells us that this child's dad is going to have to be male number 1, not male number 2. And we can check again at another band. Here's this fourth band in the child's DNA. If we look at mom, we notice she does not have this band, which means this child had to get it from dad. So we look over at the possibilities. Male number 1 clearly has the band, but male number 2 does not. And so that helps us determine that this must be the child of male number 1. And this shows those banding patterns really clearly so that you can tell every band from this child either has to come from mom or from dad. And that makes perfect sense because a child is always the combination of mom and dad DNA. Okay, so how do we do this? What does this look like? The first thing we do with creating a DNA profile or using DNA fingerprinting is we get a DNA sample. And a lot of times that's saliva, it could be semen. You can use blood, although we've talked about red blood cells don't have DNA in them, so you would use the plasma from a blood sample. Then number 2 is super important. So this one right here is really, really important. I want you to pay close attention to this. We take that DNA sample and we cut it into smaller pieces because remember, DNA is a really big molecule. It's really long. And even though it's microscopic, it's very, very long. And so we take something called restriction enzymes, which I want you to think about just as a pair of scissors. Nothing, this is just an enzyme that we can engineer that can cut DNA into smaller pieces. and it always cuts at the same pattern of letters. So different restriction enzymes will cut at different sequences. A lot of times they'll look for a GAAT sequence. And anytime they see that in someone's DNA, that enzyme is going to cut right in between the two A's. And that's called a restriction enzyme. Now, every single person on the planet is going to have some sequence of GAAT in their DNA because we're talking about, millions and millions of letters. At some point, those 4 letters are going to show up together. Any place in our DNA where that particular sequence, GAAT, shows up, the restriction enzyme comes in and cuts that spot. And what that does to our DNA is it cuts it up into smaller pieces. But everybody's pieces of DNA are going to be a little bit different because everybody's sequence of GAAT is going to happen at different places in our genome or in our DNA strands. Once we've cut up the DNA with these special scissors we call restriction enzymes, then we're going to put that DNA into a gel and run it through gel electrophoresis. And just like we've talked about before, the smaller pieces are going to go much further. The bigger pieces kind of stay up towards the top of the gel, and that electricity pushes them through. Now, we can't see DNA with our naked eye, so we're going to add some dye to it that allows the DNA to glow. And that's what this image is right here, is this picture of glowing DNA. We'll actually do that with our crime scene analysis too, so you'll be able to see that DNA sort of glowing. Now, sometimes there's not enough DNA, and if that happens, then we take a really small sample of DNA and we will amplify it using something called PCR technology. And we'll talk about that another time. PCR is short for polymerase chain reaction, and it's a way to really, really increase the amount of DNA that you're looking at. Okay, so this is a picture of what a restriction enzyme cut would look like, and I just want to show you sort of what happens with a restriction enzyme. Remember, these enzymes are designed to cut at a specific sequence of letters, and every single person has that sequence of letters, but where it happens in our DNA is a little bit different from person to person. And so what happens with these restriction enzymes is it finds this sequence. In this case, the restriction enzyme is looking for GATC, and it finds that sequence and it actually cuts the DNA apart. And you see, this would be a long strand of DNA, but what we've done is we've created 2 separate strands of DNA here. They've been cut apart by this restriction enzyme, which is kind of interesting. Okay. All right, there is a little bit of an animation here that I can play for you. Let's see if this is working here. And it may not. I may have to come back to this. In fact, actually, I think what I'll do is I will make sure that I record the link there for that animation, and I'll put that in our class materials in just a second. Okay, so we've already talked about gel electrophoresis. This is nothing new for you, but we will take the DNA that's been cut up, we will put each person's DNA into these wells, and we will run that electricity through DNA. And remember, DNA has a negative charge, so it's going to be pulled to the positive side of our gel electrophoresis chamber, and it's going to pull those particles through. Now remember, smaller pieces are going to go further. Larger pieces kind of get stuck up here. They can't move as easily. Once we have run the gel, we can look at the resulting picture. And what happens with that is that we take a look at I'm going to go backwards here a little bit. Once we've run the gel and our DNA has run through there, we have added a dye that allows the gel to glow a little bit, or phosphoresce, and then we can look at the banding patterns that we see. And here's a great example of some different banding patterns that we may have seen. And you'll notice that this DNA sequence looks almost identical to this one. And so it's possible that these two samples are from the same individual. Also, this sample and its banding pattern looks identical to this one. And this one looks identical to this one. And what we're looking for when we run a gel is exactly the same banding patterns so that we can determine who committed a crime. For our lab, the one that you'll be starting soon, we will have several suspects as well as DNA that we collect from the crime scene. And presumably, there's going to be a couple of different crime scene DNAs, or there could be. We could have DNA from the victim at the crime scene. We could have DNA from the perpetrator at the crime scene. And so what we will likely do is compare the DNA from the crime scene to our variety of suspects to figure out who in fact committed a crime. Okay, sorry about that. I've got to get through this. There we go. Okay, we've talked about gel electrophoresis. I think you understand how DNA is going to be cut up into little pieces so that we can run it through a gel and we can look at the differences that will exist. So just to give you some numbers, so you understand how unique your DNA is to you, we have these locations on our DNA that this restriction enzyme can cut, right? So this particular sequence that we're looking for. If you were to just look at one location on our DNA, perhaps our DNA would be so similar, it would be like sharing a single same digit in our social security number. So it's not very telling. It's not gonna narrow it down to a specific individual. However, if we use more than 13 different locations, like the FBI does, the odds of having two separate people who have exactly the same DNA banding pattern or DNA profile is going to be less than 1 in 100 billion. And so this ends up being a really reliable way to determine who a suspect or who a perpetrator might be. So really, unless you have a twin, you are more likely to win the Publishers Clearinghouse sweepstakes, 1 in 50 million, than to have a DNA profile that matches anyone else's. So our DNA is really, really unique, and that's an important thing to remember, for sure. Okay, that concludes our DNA profiling little video. Fill out the note sheet, make sure you turn that in, and I will see you all next time.